This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 120, recorded on January 28th, 2016. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, how are you doing? How's the snow?
Well, we've dug out. And, yesterday was pretty warm, so a lot of it melted already.
Oh, it did. Okay. No floods?
No floods. We got lots of water as a consequence. It was a little disruptive, but we're okay. Here in New York, we know how to handle snow.
That's right.
Down in Washington, D.C.,
God.
I just spoke to someone at ASM. Do you know in Washington, there's still no school?
Oh, my God.
And today's Thursday, and they said probably Friday. They have the whole week off.
My goodness.
The kids will love that,
But
right?
they had it worse than you did, I guess. It was just as bad.
As much snow as we had, but they're not really as prepared because they don't get it as often. Right?
That's right, yeah.
But New York City, we rise to the occasion. This episode of TWiM is sponsored by the ASM Grant Writing Institute online webinar. This is for graduate students, postdocs, and early career scientists. You can apply for this webinar series. It's all about grant writing. And if you're an early career scientist, you probably don't have a lot of experience at that. So you might want to take this. You'll get an overview of the grant writing enterprise, including writing NIH and NSF grants, viewing grants from the reviewer's perspective. They're the ones who are going to decide really if you're going to get it or not. They're going to score it. And so what do they look for? They'll tell you all about this. It is a 6-part series. Takes place from March through June of 2016, and the application deadline is coming up. This is the last TWiM we will tell you about this on, because it's February 10th. So if you're interested, please go to bit.ly/ASMGWO16. GWO stands for Grant Writing Online. bit.ly/ASMGWO16. We thank ASM for their support of TWiM. Elio, it's you and me today.
Okay, let's do it.
Is this the first time that it's just you and me, or have we done this?
No, we've done this once before, I think.
Okay, that's fine. I have no problem. Our other...
I'm going to lean on your shoulders.
You might. Well, we're both going to collapse then. Our two co-hosts are unavailable, so we have a snippet in a paper for you. And I'm going to hand it over to you, Elio, for your snippet. This is very cool.
Yeah. I have a story here which is totally unexpected about the shapes of things to come, the shapes of things microbial. Let me read you the title. It's an article that appeared in Science 2 years ago, actually. So it's not the latest, but nothing new has been published. So it's up to date. Marine tubeworm metamorphosis induced by arrays of bacterial phage tail-like structures. That's a mouthful. The authors are Nicholas Shikuma, Martin Pilhofer, Gregory Weiss, Michael Hadfield, Grant Jensen, and Diane Newman. All of them except one from Caltech. Hadfield is from the Kewalo Marine Laboratory at the University of Hawaii. I talked to the first author, Nick Shikuma, about it, and I'll tell you about what he told me about the discovery. All right, so the story is the following. Larvae of invertebrates in the ocean need to settle down in order to thrive, and that's true for the larvae of a worm, which is responsible for the fouling of ships, among other things. But this is a general phenomenon about the settling down of larvae as part of their development. And this involves metamorphosis. So the tubeworm, which is called Hydroides elegans, has to metamorphose in order to settle and further develop. And it does that via a bacterium, a marine bacterium called Pseudoalteromonas luteoviolacea. And this this is now a model for invertebrate metamorphosis. So the bacterium induces the larvae to metamorphose and settle down.
That's amazing.
Okay.
That's amazing.
Isn't it amazing? So, I mean, there's no end to how much communication exists between bacteria and metazoans. Is the bacteria and everything else, bacteria and bacteria. So communication is probably the theme of, isn't it? Sort of the theme of modern microbiology.
Indeed it is. Yeah.
So anyhow, so this is a story that tells you that the agent of communication is a weird, heretofore unknown molecular structure. So let me jump into that and then I'll come back. So the structure, just to give you an idea, listeners have to go to this paper because I can't describe it fully. Imagine something the shape of a cupcake made up of rods, okay, held together at the base by some amorphous structure, but also being covered at the top, like the sugar frosting of a jelly, or is made up of structures which are hexagonal, a bunch of hexagons. Can you picture that on a molecular level? Okay. And this is the structure which seems to be involved, made by the bacterium, released by the bacterium into the medium, picked up by the larvae, and they metamorphose as the result. So far, we don't know how they do it, how this works. In other words, what is it about this particular structure which makes the larvae metamorphose? Nobody knows. Okay, zero knowledge about that. But this structure, which is seen on the electron microscope, is totally new and amazing, except for one thing, that each one of the rods looks and acts like a tail of a phage. Okay. So we're talking about something that, I mean, you look at it and say, this can't be true. This is unbelievable, right? This is beyond.
The photos are amazing.
The photos are totally amazing. So let me back off. The idea that tails are used for other things than making phages is not a new one. In fact, many bacteriocins, that is, protein elements made by bacteria that kill other bacteria, are in fact in the shape of tail fibers. Okay, this is well known. They're called tailicins sometimes. They have names like that. They work by killing other bacteria by doing something that tails of phages are good at, namely attaching to the membrane and punching a hole. And this depolarizes the membrane and kills them. So this is not unknown. There is a whole collection of things that do this, and they have effects on bacterial pathogens. That is, they affect the hosts of bacterial pathogens in things like grubs, wax moths, and many others. Now, here is something which is different in that in those cases, the tail does something damaging. Here it does something beneficial to the host. That's news. But it does illustrate a principle in biology which is just lovely to me, namely the reuse of structures. When biology invents something, which it does all the time, it's not usually just done for one purpose. So here the idea, the tail fibers or the tails are not just made for making phages, but they have a completely different use. It's just very lovely. It does a lot towards explaining a lot of evolution because this reuse of structures helps understand how things evolve, I think. It's another subject.
Do you think the phage tails were first or these structures were first?
I would guess that the phages were first with tails, and then that this got adopted.
Right.
There's also even a case, I don't know if you've heard of this, but there's even a case, and I'm not sure how good it is, that capsids, that is the head of the phages, empty of their DNA, may also be reused for another purpose. And people think that there's some inclusion bodies which exist, or some intracellular structures which are like vesicles which exist in many bacteria, like carboxysomes, may have something to do with phage capsids. Have you heard of that?
I have not heard of. I've heard sort of vaguely about that.
I'm vague about this, but anyhow, the fact that nature is efficient at reusing its products, it's just a lovely thought.
Do you know the story about one of the CRISPR-Cas components is actually a viral gene. Let me see if I can remember
No
this.
kidding.
Let me remember this now.
Boy.
Here. So one of the CRISPR-associated nucleases seems to be related to a viral capsid protein.
Wow.
From a hypothermophilic archaeon. Right?
Wow.
So the capsid protein, they found homology between this Cas4 nuclease and the capsid protein. The capsid was probably first, and it evolved to be part of this defense system. I'm sure there are many, many other examples of this, right?
There are lots of examples, and I think it's just the general principle in evolution that if you invent something, you try to find other uses for it.
Yeah.
Okay, so anyhow, here's the story in a little bit, to go back to it a little bit. The genes involved in making these things had been known. They were, in fact, identified by somebody called Wang and colleagues. And so in talking, actually, to the first author, Nick Shikuma, he wanted to make sure that I made that point, that this is not the paper, theirs is not the paper where they discovered the genes. The genes have been discovered previously, also by somebody in Hawaii. So anyhow, these genes are involved in making these components, and if you look at the phylogenetic tree, they are related to others which are made by other bacteria, and in fact, not unrelated to the phage T4 GP19 protein, which is a tail protein. Okay, so these tails, which are what goes into this amazing structure, the cupcake, let's call it, for want of a better term. Can you live with that?
I love it.
Okay, so the fibers of the cupcake are the tails, and they're made up of like decent phage tails of a contractile sheath, an inner tube, base plate, and tail fibers. They're perfect, except that they lack DNA. Oddly, in the cupcakes, you find that there's a variety of them. That is, they're not all complete tails. Some are just the sheath, some are the inner tube, the inner tube. Some are, so that some lack the base plate. They're all different, okay? But the majority of them look like 70% of these structures look like a decent tail. Why this is, nobody knows. In other words, in assembly, the structure, there is some give. It's not all or nothing, okay? There are intermediates which are assembled that way. So anyhow, how did they do this? Well, let me then, there's more details, and the morphological details are fabulous. All this, by the way, was done by cryo-electron tomography. You know, the technique where you take a biological material which has to be thin. That's a problem. These bacteria are thin enough to see it. And you freeze it at minus 200 degrees or something. Liquid, is it liquid helium? I forgot. Anyhow, you plunge the material into very cold temperature so there is no crystallization made. Mm-hmm. Water stays the way it is. And you then put this under an electron microscope and you tilt. Mm-hmm. You take tilt views of it. In other words, a tomography. It's a lot like doing a CAT scan. It's really something like it. And this gives you a three-dimensional representation of material which has not been fixed.
Right.
Okay. In fact, the only the freezing itself does not do anything bad because you can take bacteria, freeze them at the temperature, and thaw them, and they'll swim away if they're motile. Okay, so they don't care about the only damage possible is the electron beam, and that's a tough one to get by because without electron beam you don't have an electron microscope.
Yep.
So. But other than that, this is about as close to reality as you're going to find in biology. Anyhow, one of the authors, Grant Jensen, is a master of this art. He's developed a lot of the technology and a lot of the things that go into it, and it has published extensively on the cryo-electron tomography of bacteria. So this is how they did it, and they saw this under the microscope. They deal in a fair amount with morphological details, but I think not having the paper in front of you, the readers may not appreciate. So we'll skip on that. So let me tell you how this happened. So Nick Shikuma, who's a new professor, a new faculty member at San Diego State, which I'm associated with, and who happens to be lecturing in a course that I organize this very afternoon. So this is backwards. I should be talking about this after listening to him, not before. Anyhow, he's. Let me tell you a little bit about him. He started doing science for a master's degree at the University of Hawaii and worked on larval development. The University of Hawaii, he tells me, is the greatest place on earth to study the development of larvae of invertebrates. So then he got a PhD at UC Santa Cruz, where he studied, he worked on Vibrio cholera. He turned microbiologist. And then he did a postdoc in the lab of Diane Newman at Caltech. Well, Diane is an amazing person, and she did something that I'm not surprised knowing her. She talked to him and said, what are you interested in doing? And instead of selling him on what she was doing, she totally took to what he proposed to do, namely this project, and says, go ahead, do it. And this is amazing. Not everybody does that. And it paid off in spades. So here's the story. This started out actually by having an undergraduate using the machine that is doing the cryo-electron tomography. And as you can imagine, at a place like Caltech with very bright students, everybody wants to do research. The easiest thing to do is to tell a student, have a look at another bug. And Nick provided him with the Pseudo-Alteromonas luteoviolacea. And the guy took pictures of it. And they had a lab meeting. They had a lab meeting with Grant Jensen and Diane Newman and Nick Shikuma and the student. And the student just casually showed these pictures. They almost dropped. I mean, they couldn't believe it. Here they see a structure which not only was unfathomable, nobody had ever seen it, but it was really incredible, really beyond anybody's power of imagination. I don't think that these cupcake-like things would be anybody could invent them. So that's the story. I think it's really a nice story. Now,
did the student know what he was seeing, or
no,
he had no idea?
Had no clue. The student just took. The student probably thought, well, that's what you're supposed to see.
Yeah.
He didn't know that he was looking at something that nobody had ever seen before. So. Isn't it nice?
It's amazing. It's great. This is the way science should be, you know?
Exactly. This is not exactly hypothesis-driven science, but nature has a way of telling you the goodies you need to know.
Yeah.
So it was just gorgeous. The pictures are amazing. People just have to go look at this. I wish we did this on television because in this today, now it's not always true, but today it's true that the visual part would be so important.
Someday we will.
Okay, it's a deal. Anyhow, so that's my little snippet. And but just to round it up, missing is how do these cupcakes work? How do they cause the development? Nobody has a clue. So this is probably in the works, and we may revisit this sometime.
Is he working on this now at UCSD?
I don't know. I don't know. He just came, so I haven't.
I guess you'll find out this afternoon.
I'll find out this afternoon.
So the bottom of this cupcake is connected by the tail fibers, right?
That's right.
And these tail fibers would be normally part of the phage itself. The phage, I guess you would call that the neck, right?
Right. That's right.
Here they're connected, which is unusual because normally they're not. So somehow this evolved to connect them, right?
Well, it's connected in two ends. The bottom end is connected to an amorphous mass, quote unquote.
Yeah, right. It's like the top of the
top
cupcake.
has these amazing hexagons. It has like a membrane made out of hexagons, probably protein, and nobody knows what that's about. So it's held, as I say, it's a little bit like looking at the frosting.
Yeah. So there must be on the tubeworm larva, there must be a receptor for this thing, I guess, right?
I think, of course.
And then that's triggering the morphological maturation, yeah.
That's right.
But it's just cool that it's part of a phage, basically, and it's evolved to have this function. It's really stunning. Yeah. I can imagine when they saw this, they probably fell out of their chairs.
It really, I mean, he tells me that their jaws dropped. That's the proper reaction.
Yeah, you don't always get to see something totally new, right?
That's right. I mean, how often do you see something that nobody's ever seen?
Right, right. No, it's beautiful. It's a lovely piece of work, and it's just very thought-provoking, and you must wonder that there are many other examples like this out there, right?
I'm sure. I'm sure there are.
Yeah.
Of course, there must be.
Okay. Thank you, Elio.
Sure.
Marine tubeworms. So these are bad for. They call it a biofouling agent. These tubeworms.
Oh yeah, ships accumulate garbage in the hulls. They're not garbage; they're living things. I shouldn't say garbage. When they point to the mussels and other invertebrates, including tubeworms, which slow down the passage of the ship because this is like having a brake on.
I presume
biofouling.
Yeah, they must get into pipes also and all sorts of things, right?
Oh, sure.
So when they settle down, they undergo morphogenesis, right? And then they get up and swim around again.
Well, I guess, sure, they must. They
go places.
They lay eggs. They lay eggs, and the eggs hatch into larvae. The larvae swim around, and nothing happens until they differentiate further and can settle down.
Very cool. All right, thank you. Beautiful. Well, tell me if he has anything interesting to say on his seminar.
I'm sure he will.
This episode of TWiM is also brought to you by the 30-second Clinical Virology Symposium. It's the 32nd one that they're holding. It's not 30 seconds long. Some people have written to me and said, sounds like it's a really short meeting. 30 seconds would be way too short. You can now send in your abstracts for this meeting, May 19th through the 22nd in Daytona Beach, Florida. So if you're thinking now you've had too much snow, you want a little warm weather, you should apply right now, even though in May it's going to be warm everywhere. Submit your abstract before March 17th, and you can present your latest research on rapid viral diagnosis, the clinical course of viral infections, and preventing and treating viral infections. There'll be over 1,000 researchers there, as well as primary care physicians. I bet some of you are working on Zika virus right now. We should send it in. You go to this meeting, and I bet you'll meet a lot of other people that are turned on by this emerging virus that's made the news this week. All right, that's the 32nd Clinical Virology Symposium. It is longer than half a minute. If you want more information, go to asm.org/CVS2016. We thank ASM for their support of TWiM. All right, our paper for today was recommended by a listener whose name is Blake. He wrote, I've been listening to TWiM for about 6 months, and I am hooked. I love how you guys challenge each other and discuss some issues you don't understand and slowly make sense out of it. I hope to hear more of these podcasts to come. Also, you mentioned Ophidiomyces ophidiocola, the causative agent of snake fungal disease. As a former graduate student, I worked with this organism and now currently working on a manuscript of the research I did. If you guys have some time, you should really take a look at two scientists that are teaming up to tackle this disease, Chris Corneliuson and Matt Allender. Dr. Corneliuson is also working with white nose syndrome and using a bacteria to treat bats with the disease. He posted some links to this work. Weather in Atlanta, Georgia has been nasty. It's been raining for several days, and I am sure Michael feels my pain. Michael Schmidt. So I was intrigued by this because, of course, I've heard of snake fungal disease. So I decided to look it up. And in fact, Blake, I'm sorry that I didn't pick a paper that was done by either Chris or Matt, but by another group. But there are a number of papers about this. So this is all about fungal diseases that are affecting wildlife species. In the last 10, 20, 30 years, these seem to be increasing in number. And many of you have probably heard of the declines, and these are large-scale declines of amphibians like frogs by what is called the chytrid fungus, Batrachocritium dendrobatis. Can you say that, Elio?
Then I would say dendrobatis.
And then the first part is Batrachocritium, maybe. Yeah. Anyway, that's a chytrid fungus killing frogs. And then, of course, white nose syndrome of bats, which Blake mentioned, caused by Pseudogymnoascus destructans. That's easier.
What a name. What a name.
Well, the bacterium in your paper was quite long also. This is the episode with long names. Now, why are these fungal diseases emerging recently? Well, one theory, which is set forth by a number of individuals, including our friend Arturo Casadeval. You must know him, right? Right, Elio?
Yep.
He works on fungi. And so fungi, his idea goes that fungi like to grow at lower temperatures and mammals have higher temperatures. And so somehow he thinks the fungi are acquiring tolerance to mammalian temperatures. And this is why they can be virulent. They can grow in the mammals or the amphibians, and they can cause disease. And what's causing this? Well, his idea is that climate warming could select for fungi that could grow at higher temperatures. They have to adapt to the increased temperatures, and these fungi are now able to colonize other animals that have a higher temperature. So now in the case of the white-nosed bats, the idea is there that they're hibernating in a cave and they're pretty cool, and that's when the fungi take hold. But in people, there have been a number of increases in fungal diseases like Cryptococcus gattii in North America. There's an epidemic of that. Fungal keratitis in people who use contact lenses. And then there's also a steroid injection-associated fungal meningitis caused by Exerohilum rostratum. And so these are examples of increasing outbreaks of diseases in humans as well, besides animals. So this paper deals with snakes. In 2006 was the first documented occurrence of population declines that are associated with skin infections in snakes. So this is like a dermatitis almost. It was first reported in New Hampshire in 2006, and then subsequently a report of similar fungal infections threatening rattlesnakes in Illinois.
You know, snakes is such a bad name. Some people are probably say, why aren't you glad that they're dying off? Well, that's the wrong way to look at it. They're part of nature. They're part of who knows what cycles. They're probably essential in their own niche.
Of course.
Let's say something good about snakes.
I think snakes are lovely. Look at them, how beautiful they are.
They are, in fact.
I mean, they have no legs and they move so gracefully, right? It's incredible. They're all colors. Some of them are poisonous, I understand, or venomous, we should say. But
and some don't shouldn't be where they are. I mean, Florida is beset by these African pythons, Burmese pythons, which are taking over, and that's not a good idea.
Yeah, but I think they're lovely. Yeah, and as you say, Elio, any wildlife we should be trying to help because they're all part of the ecosystem. It's all balanced.
Should be respectful.
Take it away, and things can happen. So this is now called snake fungal disease. It's considered an emerging disease, and it's considered to be threatening snakes, in the eastern United States. Now, the fungus Ophidiomyces ophiodicola, would you say it that way? Ophiodicola?
Yes. Yeah, sounds good.
Has been suggested to be the cause of this disease, and it's been isolated from snakes with the disease. It's an ascomycete, which means it makes spores in an ascus.
Can I say something about that?
Yeah, sure.
There is a problem with fungi in taxonomy.
Yeah.
Fungi are divided into these large groups, the ascomycetes and the basidiomycetes,
right.
The two main groups. And this is based on their sexual reproduction organs. That is, ascomycetes make a structure called an ascus, which is a bag with usually 8, sometimes 4 spores called ascospores, and they are arised by sexual reproduction. Basidiomycetes are like most mushrooms are basidiomycetes. They arise from a cell which is called a basidium, and again, sexually. Now, the problem is the overwhelming number of, not overwhelming, a tremendous number of fungi, especially what you find in the environment. Some are filamentous, molds, some are yeast, don't have a known sexual cycle. So in the past, there was a problem because they were given a name, and then somebody discovered they had a sexual cycle, and now all of a sudden, they were a different fungus. They got a different name. So that was a problem. The problem is solved in part by looking at the sequences, by looking at some of the DNA. Mm-hmm. So we can tell what group it belongs to without having a sexual cycle at hand. The nomenclature is fun. So now the mycology taxonomy committee, whatever it is, decided no more 2 names. Obviously, you can't name the same organism with 2 names, one for the asexual, one for the sexual cycle. So now there is a single name. By the way, the term for the form that is stuck in an asexual form is called an anamorph.
Anamorph.
An anamorph is a fungus which is not known to have a sexual cycle and is the equivalent of one, a cousin of one that does. That is, you say this Ophidiomyces is an anamorph of something else. That's the way you use the terms. But the term, so it's confusing. But in reality, this is one of the good uses for sequencing. You can solve this kind of a problem.
Interesting. Yesterday, our seminar speaker was Joe Heitman. Do you know him, Elio?
No, I don't.
From Duke. He works on Cryptococcus, and he's very interested in sex. And he was talking all about how organisms can have mating types, or sometimes they mate with themselves, depending on what they need. Homothallic, very interesting. If they can't find a mate, they just reproduce on their own. And in fact, it turns out he's the editor who handled this paper for MBio that we're going to talk about. This Ophidiomyces is a fungus that's only been found in snakes, and it's a member of a group that you only find in reptiles. But so it's been consistently isolated from snakes with the disease in the wild, but they don't know if it's the cause of the disease or is just something that invades necrotic tissue. Maybe the snakes are getting sick for some other reason, and this is invading it. So that's part of the problem here. The disease is, so far up to this paper, the disease has been highly variable. Sometimes it causes 100% mortality. In other cases, the snakes get better and they're fine. It's been reported in wild snakes from 9 states, including Illinois, Florida, Massachusetts, Minnesota, New Jersey, New York, Ohio, Tennessee, and Wisconsin. And it's suspected that it's much more widespread than that. It's been found in multiple species of snakes, including the northern water snake, the eastern racer, the rat snake, the timber rattlesnake, the Massasauga, pygmy rattlesnake, and the milk snake. And so lots of different kinds. The clinical signs include scabs, crusty scales, subcutaneous nodules, premature separation of the outermost layer of skin. So in other words, that's what's coming off when the snakes molt. But in here, if you're infected, it happens too soon. White opaque cloudiness of the eyes. I found lots of photographs of snakes with that. Localized thickening or crusting of the skin ulcers, swelling of the face, and nodules into deeper tissues. And it seems to vary. The signs seem to vary depending on the species. Now, aside from the presence of fungi with disease-associated lesions, specific pathological criteria for the disease have not been established. So what's needed is an animal model, and that's what this paper is developing. It's called Experimental Infection of Snakes with Ophidiomyces ophidiodicola Causes Pathological Changes That Typify Snake Fungal Disease. It's by Lorch, Langton, Werner, Fallendice, McCurley, and Blehert. They are at the university. They are at the U.S. Geological Survey in Madison, Wisconsin, and the New England Reptile Distributors in New Hampshire.
And by the way, this is interesting. I didn't know the U.S. Geological Service had a thing called the National Wildlife Health Center.
That's right.
It's interesting.
Right. So there's no university associated with this paper, as you can see here.
Oh.
These are other.
No, but so, I think it's very good that the Geological Service deals with wildlife health.
Yeah. All right. So in this paper, they use corn snakes that have been bred in captivity, and they have a pure culture of Ophidiomyces, and this was isolated from a water snake in the wild that died of the disease. It's grown in culture, and what they do is they harvest the spores, the conidia from the cultures. They wash them, they count them, so they can deliver a regular number of spores to each animal. So how do they inoculate a snake? They take each snake, and they have 8 of them that they infect. They expose them at 5 different locations: the snout, the chin, the ventral neck skin, the ventral body skin, and the dorsal body skin. And the way they infect them is they rub the skin with sandpaper, they put the conidia on a bandage, and then they tape it on. Except for the snout and the chin, they can't put anything on them, so they just sandpaper it and spread the inoculum. Now, they find that when the snakes molt, which they do, and they get better because they're shedding, I guess, the spores. So whenever a snake molted in this study, they reinfect them. And some of these snakes got infected 3 times because they molted twice. And what they find is that inoculation causes clinical signs in the snakes that are consistent with snake fungal disease. They see skin lesions in the snakes, and then they look at these macro and microscopically. They can see areas of necrosis, inflammation. They can see that the conidia, the arthroconidia, the fungus is growing on the surface of the snake and sometimes penetrating below it. They see hyphae and granulomas in these. And so there are lots of lesions in these infected animals that are very similar to the ones that they see in the wild. There is no disseminated infection. So in other words, the infections remain relatively superficial, sometimes penetrating the scaling of the snake itself, but it doesn't get into the circulation and spread throughout the snake. So, it's on the skin wherever they inoculate it, and that's pretty much where it stays. Now, they harvest, so these snakes are getting ill, obviously, and these kinds of manifestations are what you see in nature. They then recover samples from the snake skin. They do PCR to show that the snakes that they infect have Ophidiomyces at the lesions, but the control snakes, and they did have control snakes that they inoculated that just got buffer, they do not recover Ophidiomyces by PCR from them. They also sequence part of the genome of the recovered material from the snakes, part of the Ophidiomyces genome, and they show that it's 100% identical to the isolate that they used for the inoculation. And that's the only fungus that they cultured from the snakes. So nothing else grew out of these snakes, just the thing that they put in.
Does that make Koch's postulates?
Yeah, it does, I think. You got, so they originally isolated this from diseased animals. They inoculated into animals, and they got a disease that resembles the original. And they recover the inoculated material, and they show that it's the original fungus that did it. So that fulfills Koch's postulates for this. So looks like Ophidicola can in fact cause these kinds of symptoms consistent with snake fungal disease. Now, there are 2 other observations they make that are interesting. I mean, many snakes in nature die, and the question is why. We don't really know why. One thing they observed is that the snakes that were infected with Ophidiomyces, they molted more frequently than animals in the control group. In other words, they shed their skin more frequently. So somehow the fungus is triggering this. And then that's bad because, to make new skin it requires a lot of energy, and that may be detrimental to the snake, maybe makes it vulnerable to predators and so forth. And the other observation they make is that snakes in the infected group were more likely to be found in conspicuous areas of their enclosures, right? So the other snakes, the uninfected snakes, tend to hide in the cage, right? They curl up in a corner underneath a rock or something. Snakes like to do that. But these guys were just lying out in the open when the ones that are infected. So they speculate, why are they doing this? Maybe it could be a response where the snake wants to be warmed up, because this is an evolutionary response to getting a fungal infection. You warm up, maybe you cure the infection. I mean, the snake doesn't know it's doing this, but it could be some kind of response to it. But of course, if you're lying out in the open, then a predator can come over and take a bite out of you. So that could be a problem for the snake. And they also think that this fungus can affect the face. And it makes, in some of the animals here, it made the mouth swell. And they say that could affect eating. So maybe they don't get enough nutrients. And that could be also...
Could it also be that this is a socialist, altruistic mode, that is, you want to stay away from the rest of your fellow snakes so you don't infect them?
Yeah, like you when you have a cold, right?
Right.
That's right. Now, 2 of their snakes also became anorexic. They wouldn't eat. Now, what do you feed a snake? You feed a snake pinkies. These are baby mice, newborn baby mice. They like them. They just eat the whole thing, and it goes in them, and you see a lump in them, and eventually it digests. But 2 of the snakes didn't want to eat. So maybe that's also another reason why the snakes die in the wild. They don't want to eat and so forth. They also speculate that the continuous molting, the healing of the damaged skin, all this requires immune responses and energy. And so maybe, this fungus is impacting on the balance of the snake, the energy balance, the nutritional balance, and so forth. And together with lying out in the open, this could really compromise them and could account for their decline. The bottom line is now you have a model to study snake fungal disease, at least caused by this one fungus. They don't really understand how it's transmitted from snake to snake. They don't really know where this fungus is in the environment. I don't think anyone has found it outside of the snake. And so it's not clear how it would get from snake to snake. Would it be snake to snake or environmental, or maybe even some human intervention, could transmit it? Our letter from Blake said that someone is trying to treat these fungal infections with bacteria. And I'm not sure how that works. There are no papers on that yet. So maybe Blake, when he publishes his work, we'll learn more about that. Of course, even if you had an antifungal, I don't know how you would treat this, right? Because these are wild snakes. So it's not clear to me what you would do about that. So we have identified a cause. We know we have an animal model. We have a snake model to study it. But I'm not sure what you can do to prevent the spread of this disease. What do you think, Elio? I mean, how could you impact it?
Oh, my goodness.
It's tough, right?
You'd have to feed them
Antifungals,
and you'd have to feed the prey, sort of have some kind of animal that contains the antifungal.
Yeah, you could do that. If you find an area where there are a lot of animals affected, you could treat them in some way. You could give them food. But it's not easy. Yeah, not easy at all.
Well, let me ask you. I realize I know nothing about the snake immune system. At what point does the acquired immune system evolve? Do snakes have antibodies?
What a good question.
Let's look it up.
I'm going to look it up because I don't know. Snake antibodies.
Because this would matter in terms of what lifestyle, what kind of intervention you would dream of.
Yeah, they have antibodies. Yeah.
Yeah. Okay.
Antibodies are invertebrates, and snakes are vertebrates. Yep.
Right.
But people have studied them. I find a paper here on snake antibodies.
Right. Well, so you could think of somehow having a vaccine.
Yeah. How would you get it to the snakes? You'd have to drop feed with the vaccine in it, right? Yeah.
The live vaccine, and that's okay. Live vaccines in the world.
Well, we immunize wild animals against rabies by dropping a bait into the woods that has the vaccine in it.
There you go.
And the wild animals, and it makes an impact. It does immunize a certain fraction of wild animals. So you could do that for snakes. In the wild, snakes eat.
Go over herd immunity.
Yeah, so that's right. You get a certain number of snakes immune, and then that would presumably knock down the total number of cases, assuming it's spread from snake to snake. And there could be an environmental reservoir.
That's true.
Case, and then that's a problem, right? And herd immunity doesn't help. But snakes typically eat rodents, right, out in the wild. And so you could put rodents. I have an idea, Elio. Nobody will like this, but you make transgenic mice, which make an antifungal, and you release them, and the snakes eat them, and they get cured. And nobody would like to do that because no one wants to release genetically modified animals into the wild,
Right.
Anyway, that's the paper. You like that?
Yeah.
It's something different.
Paper.
It's something different. It's relatively straightforward. A lot of very nice photographs of 6 snakes and the kind of histology that you see. They do sections. They sacrifice some of these animals and do sections. But now they have an animal model. And I should point out that in looking up background for this paper, I found other similar papers where authors have done similar experiments in a different kind of snake. And one where I found in PLOS One, they did it in cottonmouths, a different kind of snake. And in fact, that's the paper by Allender and Cornelissen, who that Blake mentioned. But I didn't do that paper, but that's a PLOS One paper. Basically the same thing. They infect cottonmouths with the fungus, and they develop disease, and it's a similar conclusion. So I'll put both links in the show notes. Anyway, I thought that was cool. I always wanted to learn about snake fungal disease, and here you go. Here we have it.
Terrific. Nice job.
Now,
I enjoyed hearing this.
We don't have any emails, so that's it for this episode. That may be the shortest TWiM on record, but that's okay. Sometimes people like to hear a short episode. And this one you'll be able to find at iTunes, at microbeworld.org, or at microbe.tv/TWiM. And please send us your questions or comments to twim@microbe.tv. I want to thank you, Elio, for joining me today.
My pleasure. Nice chatting with you. Always is.
Always. You can find Elio at his wonderful blog, Small Things Considered. When do you publish? Twice a week at STC?
Twice a week. We have not missed. It's our 10th year. And like I like to say, blog years are like dog years. So 10 years is a long time.
And you do
We have not missed a single Monday and Thursday post.
Monday and Thursday. So which one is the long one?
The Mondays are the long ones and the Thursdays are the short ones. We have things like Talmudic questions on Thursdays. And by the way, I can't thank enough the staff at ASM. Ray Ortega has been fantastic in helping us out and doing yeoman work about this blog. There's more to a blog than I knew, and so I'm glad that he's on board.
It's very nice.
It wouldn't be the same without him.
Looks very nice, and you can find it at Schaechter.asmblog.org. And today's article is called "How Can Microbes Contribute to the War on Cancer?"
Well, we thought it'd be timely. See, you hear about the war on cancer that President Obama has announced, and the Vice President Biden is supposed to lead a moonshot-like thing.
Right.
And, history repeats itself because quite a while ago, another president, his first name was Richard.
That's right.
Proposed the same thing, war on cancer. And he predicted, and somebody in the government predicted by the year 2015, cancer would be licked. Well, hasn't quite happened. So we thought, just to make notice of this, it would be fun to see what readers of the blog come up with when asked, what microbiological aspect would you advise Vice President Biden to look into? We know that there are things like oncolytic viruses, which have been tried. Some bacteria have been tried as homing in on cancers. What else? So I would love it if readers of the blog would raise to the occasion and send us in their views of how microbiology belongs on that particular front.
Yeah, it's a good idea. All right. So I also have been blogging. I've been blogging for 11 years, which is 110 years total. And because what did you say? A blog year is like a dog year, right?
That's right.
I enjoy that very much. You can find my blog at virology.ws.
And a great blog it is, by the way.
Not bad. I enjoy it. I try to post every week. I just do it myself, more or less, and I try to do one post a week, and sometimes two. And this,
we have a team. I'm the charge d'affaires for foreign affairs on the blog, but a lot is written by other people.
Yeah, that's nice to have a team. Takes a little pressure off, right?
That's right.
Yeah, I like that. I'm a sole proprietor here, but that's okay. But I have teams on my podcasts, as you know.
And how?
Which is great because I couldn't do it without my team, including the ones here on TWiM. I would like to also thank the American Society for Microbiology for their support of TWiM, in particular Chris Condayan and Ray Ortega. They do all the behind-the-scenes stuff that we really appreciate. So thank you, guys, and thank you, ASM. And the music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.